Okay, thank you so much for coming back to Making It Brain for the second day. I really hope you enjoyed yesterday. So just to recap, if you missed yesterday's session, we had two amazing talks from Professor Nick Fox and Dr. Sarah Marzi, who are two researchers in the UK DRI. And that webinar was recorded, and we will be sending that to you in an email tomorrow, along with the recording of this session too. So thank you so much for joining us again today, for giving up some of your afternoon to join us. Today we have three talks, and as you will see, it's a really wide range of research that goes on in the UK DRI. And I just want to take this opportunity to thank also the people who work in this Dementia Research Institute who don't do lab work or scientific research themselves, but help events like this to go on. So I've worked with people from the communications team in the Dementia Research Institute who also themselves may have scientific backgrounds, but ended up working in communications. So really thankful to Alex, Amelia, and Liz, who's also on this call, for helping me run this event. So I'm really excited to announce, as I did yesterday, that we have a prize draw at the end of this session for 2 memberships to the British Neuroscience Association, so 2 A-level and undergraduate memberships. And at the end of the session, I'll also be detailing resources that you can use if you're interested in going into this kind of career or this kind of this kind of studies. So as yesterday, please do feel free to leave your questions in the Q&A box, and I will save them all for the end of the session, where we will put the questions to all of the speakers together. So, and we'll do that at the end of the hour. And after we hear from Professor Selina Wray, who is speaking first, we're going to have 2 follow me around the lab videos. One is from Imperial College London and one is from Cambridge, where I am based. So my introduction as well, my name is Sophie. I'm a PhD student at the UK DRI in Cambridge. And yeah, that makes me very excited to introduce Professor Selina Wray, who is our first speaker for today's session. And it's been really, really great fun bringing in all of these speakers, and I'm so excited to have you here. So thank you so much for coming to speak to us, and yeah, I will see you later. Oh, Selina, you're just muted right now.
There we go. Thank you so much for the introduction, Sophie, and it's really nice to join you today, and really nice to see where you're all from. I did see at least one person from Yorkshire. I saw someone from Sheffield there. So I'm going to give you a special little shout out because I'm from Barnsley, just next door. But of course, wherever you're from, far and wide, it's really great to join you today. So I will speak to you for the next 15 minutes or so about some of the work we are doing trying to model Alzheimer's disease in the lab. And particularly we're using stem cell technologies, which I'm sure that some of you have heard of, and I'll tell you a little bit more about how that's really being transformative for us. But first of all, let me tell you a little bit about myself. So I've kind of put my little academic timeline here, and I've also just wanted to highlight this. So I was actually made a professor last year, but that's not the bit that I wanted to highlight. I wanted to really tell you a little bit about my background, just to say that really scientists come from all walks of life. You will have seen that represented in the speakers yesterday and today. And my own background is really one where, you know, I grew up in a house where I was the first person to go to university. We had free school meals and we grew up on benefits, and yet it was still possible for me to have this pathway. And there are lots of opportunities out there. If anybody's listening from a similar background, there are lots of opportunities out there for support and for university scholarships to help with fees, for paid internships to help you get lab experience. And so I would encourage you to not be deterred and really look at what schemes are available. And of course, I'm really happy to kind of chat with anyone who might want more help or advice about that. I decided fairly early on that science was the thing that I'm interested in. As you can see here, my A levels were biology and chemistry and physics. And as I was deciding what to apply for at university, really I decided that I didn't much want to continue with physics. I found it a bit too hard, and you know, I really liked the kind of biology and chemistry aspects of my courses, and I couldn't decide which one of those to give up. And it turns out I didn't need to, and that's why I thought, well, I'll pick the course that combines the two of them. And so I actually went to the University of Nottingham, where I did biochemistry and biological chemistry. And this essentially was a course that was half biology and half chemistry. As you can see here, it was a 4-year course rather than a 3-year university course. And that was a tactical decision on my part, because having that extra year meant that you got more practical experience in a laboratory setting. And that was really what I wanted to get out of my degree. And I think, again, you know, I would really recommend that to anyone who's maybe thinking about a career in science, because learning about science and doing science are really two quite different things. And the only way you will know if you enjoy lab work is to try it out and get kind of as much experience as you can. During this, I was also able to do kind of a summer project in a research lab, working on Parkinson's disease. This is where I first decided that I wanted to work in the area of neurodegeneration. That was a paid internship funded by the Wellcome Trust. So there are opportunities out there to get lab experience and to be paid for that. And so having really enjoyed my lab experience, I then went on to do my PhD at King's College London. And this is where I started working on Alzheimer's disease. So I did a 4-year project working on the protein tau, which I know you would have heard about yesterday. And that was where I, you know, again, further developed my love of research. And I really liked leading and being in charge of my own project, and also being able to do something over a long period of time where you can really get your teeth into it, and kind of follow the interesting findings. After I finished my PhD, I moved to UCL in 2009, and that's where I've been ever since. So I joined as a research fellow under the direction of Professor John Hardy, who I'll tell you a bit more about in the science bit of my talk, and eventually managed to establish my own group. So John is a geneticist. He's interested in the genetic basis of disease, whereas I'm a molecular neuroscientist, which means to me I'm more interested in kind of the cell biology of the disease. So I will kind of move away from my background now to talk a little bit more about science for the rest of my session. And as I mentioned, I'm a professor of molecular neuroscience, and I thought it might be useful to start with a very simple slide to just give us a bit of orientation of what that means in the grand scheme of us trying to understand and treat disease. And so molecular neuroscience is really quite just a fancy way of saying we try to understand the small stuff, the DNA and the proteins. I'm particularly interested in the proteins that come together to make our cells, which together make our organs, which all together will come together to make us. And I showed this kind of very simple scheme because I think it illustrates very nicely that if something goes wrong at this very molecular level, the impact of that will then be felt at each step of increased complexity. And by extension of that, in my opinion, if we really want to treat Alzheimer's disease and other diseases, this is where we need to focus our attention. Because by the time things are manifesting themselves here, when we see brain damage, for example, it's already a bit too late. We really need to intervene at this molecular level. And so the question is that my lab is interested in is, why do the cells in the brain of someone with Alzheimer's die? What causes that cell death to happen? And so you will have seen something a bit like this yesterday. This is what the Alzheimer brain looks like at post-mortem. You can see that there's been a huge loss of cells as they've progressively died over time. And when we look a bit more closely at the brain, we see two protein accumulations that are really the hallmarks of the disease. The first are these plaques composed of the protein A beta, And then the second are these neurofibrillary tangles, which are composed of the protein tau. Now, I spent my whole PhD working with postmortem tissue, and it's obviously a really valuable resource. But the disadvantage of working with tissue that has been donated after somebody has died from a particular disease is that we're really at the end stage. And I always think about this as being a bit like, CSI. You turn up to the scene of the crime, but the damage has already been done. And through looking at tissue, it's really difficult to put together a sequence of events that tells us what order things went wrong to arrive at the point of having disease. And so what I'm particularly interested in is actually, can we build models in the lab that will help us follow the disease in real time? And by using those models, we can put together a kind of timeline of disease where we know what the first thing that goes wrong is and the kind of cascade of events that will eventually lead in cell death. The one way that we do that is by using genetics. And I mentioned earlier that I would talk a little bit about John, but I think it's kind of really nice as well, and you will have heard this from Nick yesterday, to acknowledge the patients who have really enabled much of the research that we do. And really the lady shown in this picture here was instrumental for most of Alzheimer's disease research as we know it now. She wrote a handwritten letter with a handwritten version of this family tree to John and Martin back in 1990 and said, well, look how often Alzheimer's happens in my family. This can't be changed. You need to do something to kind of help us understand this. Now, everyone in this family tree who's coloured in black had an early onset Alzheimer's disease, by which we mean they developed the disease perhaps age 40 or 50. And through working with these families and kind of understanding the genetics of these families, the first Alzheimer gene was identified. And what this allows us to now do is build models around that genetic finding. So a gene tells us that, if something goes wrong with that gene or the protein that the gene codes for, that's enough to cause disease, but it doesn't give us any more understanding than that. We really need to understand why that gene is so important in the context of a whole cell or a whole organism. Now, of course, we've been at a real disadvantage when we try to understand diseases that affect the human brain. And the reason for that is, although we can study the brain at postmortem, the human brain is really inaccessible to us during life. So we can take people who carry these mutations and, we can use brain imaging to visualize their brain and how it changes over time, but we can't easily take a brain biopsy during life at multiple occasions. People are generally, understandably, not very keen for that. So what has traditionally happened is we look to other organisms such as the fruit fly or the mouse, where we can introduce the gene of interest and look at the impact that has on these organisms. But of course, that also has a big disadvantage because the brain of a fruit fly is not quite the same as the brain of a human in size, in complexity, in lifespan. So what we're interested in is, how can we really make a model that is physiologically relevant? Is there a way that we can grow human brain cells in the laboratory? And the answer to that really comes from stem cells, which are shown here. I should say before I move on that although I'm very kind of passionate and very pro using these humanized models, we actually need all of the models that are shown in this image. Every model has its advantages and disadvantages. We can talk about some of those in the Q&A, and we really need to use all of the tools we have available and kind of fit that information together like a jigsaw to fully understand the disease. But the other challenge we also have is the timeline in which these changes are happening. So this is a video provided from a collaborator of mine, where you can see sequential brain scans that have been taken every year in an individual who carries the genes that we're interested in. And hopefully you can see kind of the change that's happening here. But what's interesting is that this is all happening pre-symptomatically. And that reinforces my point from earlier that we really have to use models which let us understand the very, very earliest changes that are happening so that we can intervene in the disease process as early as possible. This I already mentioned, that we need to have early models and human models because of the differences in the human brain compared to other organisms. So now let me tell you a little bit about how stem cells can happen with that. So I'm sure that everyone who's in the audience has heard of stem cells, but I think it's really useful to have two very basic definitions so that we're all kind of on the same understanding moving forward. So stem cells in my mind are characterized by two properties. The first one is self-renewal. And this means that a stem cell will divide to form two identical stem cells, each of which will divide to form two identical stem cells. And this process will continue indefinitely until those stem cells are given a signal to do otherwise. And that's when the second property comes into play, which is the property of pluripotency. I always struggle to say that because there's so many P's. But pluripotency is a translation from the Latin for most potential. And what this means is that a stem cell is a blank slate. So within a stem cell, there is the potential to become any cell type of the human body. And this includes neurons. So we now have an opportunity, if we can grow stem cells in the lab, to grow human neurons and study them while they're alive and as they age in culture, which is something that we're not able to do otherwise for the human brain. Now, this is already quite remarkable. But about a decade ago, this technology, there was a real breakthrough in this technology, which was transformative in our field. And this was the development of something called induced pluripotent stem cells. Now, when we look at this schematic, the dogma for a long time was that a stem cell would become a specialized cell type, but that was a one-way process and it couldn't be reversed. What this breakthrough showed us is that that's not the case. It's actually possible to change the identity of one cell type into another cell type using tricks in the laboratory. And that's exactly what the scientists who first described this technology did. They took skin cells, which are very easily accessible. We can take them from a biopsy in someone's arm and we can grow them in the lab very, very easily. But when what we can do is reset their identity and we can change the skin cells into stem cells. And so we induce this property of pluripotency, which is where the name comes from. Now, because those stem cells are a blank slate, we can then turn them into neurons. The reason this was such a breakthrough is because it means we can take a piece of skin from any patient who has a disease or a gene of interest, and we can use that in the lab to make their neurons. And for the first time, this means that we're able to make patient-specific human and neuronal models of dementia. So we kind of refer to these as dementia in a dish models, and they've really transformed the field. The group who first described this technology were interested in cardiac disease. And I just wanted to show you a quick video, if it will play. These are some cardiac cells that they made from stem cells in the very first description of this technology. And what you can see there is, even though they're grown in a lab, they're beaten in a dish, which I absolutely find phenomenal. But we're not to be outdone in neuroscience. And actually, this is a skin sample, a biopsy we took from one of our Alzheimer patients who is actually seen in clinic by Nick, who you heard from yesterday. We used that to establish their skin cells, and we can freeze these and revive them whenever we need them. And then using these skin cells, we made their stem cells, and we made their neurons. And these are neurons that we've grown in the lab from somebody with Alzheimer's disease. I think they're absolutely beautiful. They look like a firework that just exploded. And to me, it's really astonishing that we can grow brain cells in the lab from somebody who also comes to clinic for MRI scans, for biomarker studies, so for CSF, and actually who were monitoring their disease in life. And now we're monitoring their disease in the culture dish in the laboratory too. And it's not enough that they just look like a neuron, they have to behave like a neuron. And here what you can see in this video is every time one of the cells flashes, what we're actually seeing is them sending a signal to another neuron within the dish. So that they're not just taking on the shape of a neuron, they're really behaving like a neuron. And what this means for us is we can take cells from people with and without dementia, and we can compare them in the lab, and we are monitoring how they become dysfunctional in real time. So that will allow us to really identify the very earliest things that are going wrong and design methods to hopefully intervene in that could eventually be used in patients. So we can do drug screens in the cell culture dish. And I think I have just one more slide. Just to kind of show you some data. You would have heard a lot about A-beta already yesterday. What you can see here, these are patient-derived neurons that we've grown in the lab. These are from people who do not have dementia, and these are from people who carry these dementia-causing mutations. And you can see that already within a few months, they have a very, very different disease-like profile compared to the healthy neurons. So this works. I know it sounds a bit science fiction, but it really does work, and it really is transforming the way we understand disease. So I will stop there. I summarized what I've said on this slide, and I just wanted to echo again the point I made at the beginning, that none of this would be possible without the patients and their families, and particularly the families with the genetic causes of disease are, really special. We ask a lot of them, and they participate in research really enthusiastically, and I find that very humbling. So I'll stop there. Thank you.
Thank you so much, Selina. That's such a really interesting insight into stem cell research for dementia research. So please do feel free to leave your questions in the Q&A box, and Selina will be back at the end of the hour, and we will be answering them live too. So we're just going to take a quick interval where we're going to take a trip to the UK DRI Cambridge. So you're going to visit me here, and then we're going to go to the UK DRI at Imperial, where they do research into sleep. So lots of different areas of dementia research. So first, we're going to go to Cambridge. And now we're going to go to Imperial.
Hi, welcome to the UK DRI Care Research and Technology Centre. Come on in.
Okay, so thank you so much for leaving your questions in the Q&A box. We're super excited next to introduce Dr. Dayne Beccano-Kelly, who's here to talk to us today about the synapse, which I believe is part of the A-level biology curriculum. So I think they will be grilling you today, Jane. So thank you so much again, and I'll see you in a bit.
I'll look forward to it. I'm glad it's on the curriculum. It should be on the curriculum. It's very important. Hi. Hi, everybody. I'm hoping you can now all see my screen. And brilliant. Thank you so much for the invitation to come and speak to you today. It's really a pleasure to talk about science with what I hope will be the next generation of scientists. And so I want to talk to you about my area of research, which I conduct here at the UK DRI Group in Cardiff University. And it's specifically around the way in which the cells of the brain, the neurons of the brain, communicate through this electrical chemical interaction, and how looking at the earlymost changes in this communication could point the way to understanding Parkinson's disease, and indeed could help us to prevent it if we can understand the targets there. Hopefully keeping narcissism to a minimum, and hopefully in keeping with everybody else, I'm going to talk to you first, however, a little bit about how I reached where I am now. So it was quite interesting hearing Professor Wray's journey, because I too took a number of A-levels, of which they were chemistry, biology, and physics, and I too dropped the physics as I moved on, but I eventually went to the University of Leeds to as well do my undergraduate degree in biochemistry. And after my first year, because of my grades, I got offered the ability and the chance to switch from just straight biochemistry to biochemistry with industry. And I leapt at the chance, not least of which because of the location, which was actually at the Mayo Clinic in Jacksonville, Florida. Now, this was an absolutely fantastic placement to do and to take on, and I would highly recommend placements to get a feel of what you can do with science. But it wasn't just fantastic because of the location, it was absolutely brilliant because of the scientists that were there. And indeed, one of those scientists was my mentor and my supervisor, Dr. Desmond Stickson, who was one of the foremost neuropathologists in the world. And he really had a passion for science, a passion for understanding the clues that had been left behind as a result of neurodegeneration. And he had a fascination, which I did too, with understanding what we didn't understand. He took pleasure in the fact that we were at the edge of knowledge, and we don't understand everything that's going on with the brain. And it's quite interesting to be in a job, in a role where we're constantly finding things out. And so I knew and I realized that once what I wanted to do with my degree was to go back and finish it, obviously, and to stay in academic research, but specifically to work in neurodegeneration because I found it so fascinating. And so at, again, the University of Leeds, I took up my PhD, and it's here that I learned to perform a technique known as electrophysiology. And electrophysiology is a technique that is very important in understanding and being able to measure communication between neurons. And I'll talk about that a little bit later. And after this, I took a fairly circuitous route to where I am now. I ended up doing a postdoc at the University of Dundee at Nine Wells Hospital, looking at Alzheimer's disease models before then moving over to the University of British Columbia in Vancouver in Canada and working on genetic models of Parkinson's disease. And I stayed there for a number of years before then coming back to the University of Oxford and starting a career development fellow position, where I worked on and utilized and learned to record from induced pluripotent stem cells, which you heard a little about earlier. And it's really quite a powerful technique and platform, and something that I'll be talking about and discussing later on. And earlier this year, I won a UK DRI Future Leader Fellowship Award, which will allow me to start up my work on Parkinson's disease and looking at this synaptic communication and how it may point the way and lead the way to a cure. And so one of the things about moving around a lot is that it wasn't frowned upon. It's one of the very few career paths that you can actually move around in, and it's not negatively going to impact your work. It's not going to be thought of as being detrimental. And in fact, it's actually thought of as being beneficial because historically, each university normally has its own particular focus, its own particular niche. And in going to these different institutions, what you can end up doing is actually taking on these different ways of approaching a problem and indeed trying to then gather all of that up. So when you come to working on something, you can use a more holistic approach, trying to answer the problem from multiple different points. And it's this that I'm trying to do in my research now in Parkinson's disease. So as many of you will know, Parkinson's disease is a disease and a disorder associated usually with older age groups in older age. So on average, it usually presents as a motor dysfunction where you have this stooped posture and this central rigidity, a trunk rigidity, as well as the hallmark resting tremor, so the shaking that everybody knows is usually associated with Parkinson's disease. And this normally manifests about 65 years old. However, less people know that the only way to actually definitively diagnose Parkinson's disease at the moment is to do that after death. So to look at inclusions and things within the brain that indicate exactly what the patient has. And this is a microscopic image of proteins that have begun to stick together. And these proteins specifically are known as alpha-synuclein, and they clump together and come out of solution and form these huge tombstones to the cell inside the brain known as Levy bodies. What also you can look for is the destruction and the loss of specific cells that release a chemical known as dopamine and are really quite important in controlling movement in the body. And these neurons, which you can see here, are black, and actually they do appear this color in your brain. They have a specific pigment, are specifically damaged in Parkinson's disease. And you can see later on in Parkinson's disease, they're almost completely gone. And whilst there's been absolutely fantastic, tireless research that's yielded a number of targets that actually are very important in Parkinson's disease, there is still, as yet, despite decades of intensive research, no known interventive therapeutic that can completely halt the progression of Parkinson's disease. And I believe that this is due in part to focus on the late stages, that very end stage of the neuronal loss, the cell death. So what can we do to counteract this? Well, there's much work that has now gone into to looking at the about earlier in terms of the timeline of the disease. And so at diagnosis, when people start to present with these motor symptoms, one of the things we now understand is that before the motor symptoms actually start to present themselves, what we can see is non-motor symptoms coming about. Things like anxiety and problem-solving skills start to manifest a few years before the motor skills actually start to show up. At diagnosis, we know that those neurons, those dopamine neurons that release that particular chemical and are important in movement, are actually, we've already lost about 60 to 80% of them by the time patients come in to see their doctors. But actually the initiation, the start of that cell loss occurs at a much earlier time point. And so we know that if we find and more data and find ways of identifying the disease at earlier stages, then we can probably intervene and prevent and delay the diagnosis and indeed the manifestation of these motor symptoms. But what a lot of people are now trying to do in the field, myself included, is to actually look even earlier still, to try and look at this time point of the disease before cell death is actually prominent, and trying to look at the dysfunction that precedes that initiation of the cell death. And so what I want to do, as well as is to look at the timeline of the disease. And what I like to liken it to is actually to try and understand it a bit like how a car breaks down and how the engine starts to break down. What we want to do is we know that there are various different things that we can look out for that might tell us that the car engine is about to break down. What's important is not only to understand exactly what to look for, but it's specifically when to look for it. If we understand the order that these things come in, then we can start to look at what's associated with what and look to go to a mechanic before the engine actually has to break down. And it's the same in Parkinson's disease. We will have all sorts of different things that start to begin to slightly shift away from the normal that may give us an indication and lead to the neuronal death. And it's this that we really don't understand in Parkinson's disease at the moment, how the timeline of the disease works. And so where I think is best to look is actually at that specific thing that makes a neuron specialized, makes it specifically gives it its only physiological role, which is the communication via this chemical electrical activity. And this is a neuron inside tissue. We've managed to impregnate it and fuse it with a particular stain. This one is a silver stain. And in doing so, we can see the architecture of the neuron. You can see the cell body here, and then these projections, which can be called neurites within the system. If we zoom in to these arms, these finger-like neurites, you can see that you have these sort of knobbles on them. And these are boutons. And what normally happens is that each individual neuron has hundreds and thousands of these different boutons. And actually, as electrical activity moves down these arms, it will reach these boutons. And as it does so, it will release neuron chemicals from the system. And as you release as you release chemicals from one side of the bouton, it will travel across the gap between the two, which is known as the synapse, and then hit the postsynaptic element of the bouton from the other neuron. And this is then transferred and changed into electrical signal. And so it's this communication that is fundamental to actually do everything that we do. So thought and speech and movement, and it's all critical. And so this communication and this talking between the two neurons via this chemical, via this, these boutons, is critical to making everything function correctly. And like any particular action and conversation with an instruction, you need a couple of things to be there in order to be for it to be enacted. First of all, you need the command to be spoken correctly and accurately. And secondly, you need it to be heard accurately. And if this doesn't happen, what you'll end up with is a miscommunication, which will lead to an inaccurate response to what's actually going on. And so to measure this activity, what we need to do is we need to have a setup in order to be able to measure that. And that for us is electrophysiology. And this sounds very fancy. And it has, the setup that I use has lots of dials and wires and computer setups and tubes and pumps and dishes and lenses, but actually what it all boils down to is just having an open circuit into which you can plug your cell and then activate it, a bit like a light bulb in a circuit. You can screw it in and then effectively learn from it once you switch on and off the cell. And so to do this, you will put your cells inside this little white dish down here, move your electrode down. And actually what you can do is you touch down to the surface of the cell. And just at the point at which you touch it, you can provide a small amount of negative pressure or suction onto the cell, forming a very tight seal. And just at the point of contact, you can apply a little bit more suction and just rupture it just at the point of contact, leaving the rest of the cell intact. And in doing so, you now have the ability to allow electrons to flow through this, this gap, and actually in through this gap, and then back out through the cell, but only via these gateways, what we call channels, ion channels inside the cell. And so now inside this system, you can measure a lot of different possibilities and lots of different connotations. And one of those is called spontaneous excitatory postsynaptic current. And this sounds very fancy, but I'm going to try and explain it to you now. It's basically normally what you would have is the boot on one side and one neuron would have electrical signal, and as soon as it reaches the edge, it would cause a rush of neurotransmitter to move across the gap and then received by, in this case, the sync, which is the boot on the opposite side on the other cell. Once it is, it's transduced and the signal moves on. But in between these rush of neurotransmitter, what we actually get is that the tap is leaky. And every so often, without the signal, you'll actually have a little droplet of the chemical moving across the synapse. And when this happens, every time it hits the postsynaptic boot-on, the boot-on on the other neuron, it will cause one of these channels to open and close. And what will happen is in this mode, whilst we're recording, we'll see the opening and closing of these as these little spikes. And when we analyze these spikes, we can tell a lot of things about the neuron. If you measure the size of it, we can actually tell you how many of these little channels there are on the opposing side of the synapse. If we actually count them up, we can actually work out a couple of things. First of all, it might be that the number of these synapses have actually changed, but it could also mean that the driving force behind releasing this chemical has altered. So there might be a change in the amount of driving force there. So using this technology, what we wanted to do is to actually look into genetic mutations of Parkinson's disease to see if we could actually look and identify any changes at the synaptic level. And as mentioned before, we can see that about 10% of all Parkinson's disease is actually caused by genetic mutation. One such gene that can have a mutation that causes Parkinson's disease is the leucine-rich repeat kinase 2 or LRRK2 gene. And this codes for and creates a protein that has lots of different sections to it. And each of these sections, as you might imagine, actually results in a lot of different functions for this protein. But a lot of these functions seem to cluster around that all-important function of a neuron, that electrochemical signal, the crosstalk between them. It can seem, LRRK2 seems to be involved in the size of the synapse, involved in the movement of proteins to the synapse, and indeed the release of chemicals in the synapse. So what we wanted to do was to compare a healthy set of neurons with a set of neurons that has a mutation in this gene that is known to cause Parkinson's disease. And so when we measured that drip from one side of the synapse to the other, we found that the amount of dripping that was happening was significantly increased in the disease state. So there was an increase in activity. Now, as I mentioned, this could be a change in the number of those boutons, or it could be a change in the driving force of the activity. And actually, when we measured the number of boutons just simply by counting them, we found that there was no significant difference in the architecture. And so we knew that this was a driving force alteration, a driving force change. And so this can be quite abstract even for other scientists, but we can see the actual change in what this would mean when we look at behavior of animals. Now, we can take mice, and mice are incredibly clever creatures, and we can put them in a large open space with a light above it. When we do that, I can tell you they don't particularly like that because they think they're going to be predated upon. They think there's a big bird that's going to come in, swoop in, catch them, take them away. So we create a dark, safe space for them, and in which we have a small doorway which they can run into. And what we did is we placed them inside here and allowed them to run in and measure the time for them to be able to do this. But then we changed and altered how easy it was to get into the door. We put things in the way, and we made it a little bit harder for them to pull these things out of the way, but they were very clever and they would always work it out. However, when we measured how quickly they could understand how to do this task, we found that the Parkinson's disease mutants seemed to work it out at a much faster rate than the healthy controls. And so this all might seem a bit counterintuitive. We seem to see increased activity and increased cognitive capacity, which I told you at the beginning was seemingly down in Parkinson's disease, less cognitive activity, less electrical activity. This is not a new concept. Normally what you would have in a healthy individual is a decreased neuronal activity over time to varying scales and degrees and rates in different people. But in Parkinson's disease, from what I've shown you, we're starting with quite a hyperactive system. And the cells of the body are very good at keeping things in an optimum level, in homeostasis, and are good at adaptation and compensation. So when it sees this high level of activity, it's going to be like this too much energy. This is too much expenditure, pull it down and rein it in. And it will eventually put it down into the normal levels of activity. The problem about this is that this will only be for a short-term fix. And so what normally would then happen is, because it's consistently going on, you probably end up with a collapse of the system. And so if this is actually true, what we should see is there would be high level at young ages, but at low levels at older ages. And indeed, as I showed you before, at young ages, we had this high level of activity increase, which was associated with no changes in the number of synapses. But when we went to the older age groups, much older, we saw a flip of this effect. And so the properties in looking at the timeline of the disease seems to imply two phases to the disease. And this can be really important in understanding exactly what to do and when, because if we were to try and dampen down the electrophysiological activity at the late stages, this might prove to be even more detrimental. And so following the fantastic talk by Selina Wray is beneficial to me, because one of the things that I wanted to go on to do was to look in stem cells, because Parkinson's disease does not affect mice. And so taking skin biopsies, usually from hole punches just underneath the arm, we can drive them towards being stem cell-like. And from this pluripotent state, you can push them towards being effectively baby neurons and then onto adult neurons, mixing them with other cells of the brain to effectively create a brain in a dish. And we can take our electrode and patch them in cells. And now we have the ability to see that these neurons are active. They fire regularly, increasing that quantal drip and that regular firing activity in the brain. And so what we can now do is take skin cells and biopsies from patients with Parkinson's disease and compare them to those from healthy individuals. And so measuring that drip, that activity state in these human neurons, what we found was that there seems to be lower levels of activity in the cells from Parkinson's patients. And this seems to map up with the late stage symptoms that we see in the mice. And so with that, we can also start to measure other components of electrophysiology to really understand what's going on with the building blocks and how they all go together. We can see that the amount of input that we have to put into a cell for it to fire is actually dramatically increased, almost doubled in the Parkinson's disease lines. And in fact, when we want to get them to fire repetitively, which is quite important in coding inside the cell, it seems to be quite hard to do that in the disease cells. They seem to be hard to fire repeatedly. And so overall, what I've hoped to show you is that as a result of genetic insult in this particular case, if we measure the cells over time, we can start to see that we get changes in electrophysiological activity, in synaptic activity, and this has a knock-on effect to the behavior of the animals. But we are going to continue to look to see if we can find other components that then may specifically and directly activate or inactivate these changes that we see early on in an effort to try and find things that we can target and prevent that late stage neuronal death and indeed the presentation of Parkinson's disease. So with that, I would like to thank all the people, my mentors and colleagues and that have helped me to get all this data, because I would not have been able to get it alone. Science is a very collaborative effort. And I would like to thank my funders at the UK DRI and indeed you all for listening. I hope there are a few questions because I would love to take them.
Thank you so much, Dayne. That was fascinating. And for someone like me who's never done electrophysiology, I think that was really made it sound very, very cool. And I think a lot of people listening might be upcoming electrophysiologists. So we're now going to have our final talk of the session from Soyon Hong, who worked with me to organise this event. So I'm really delighted to introduce her today. And please keep leaving your questions in the Q&A box, and we will come back together for a live Q&A afterwards. Thank you.
Okay, great. Thanks, Sophie. It's been a fantastic meeting, and I think this is amazing. I would love your feedback so that we can, when we host it again next year, hopefully, then, we know how to improve it. So hopefully our wish is that, these, few of you out there will be really motivated to study dementia in different aspects. And, I'm very excited to tell you today, changing gears a little bit, because I'm going to talk about another type of cells that stay and live in our brains, the immune cells of the brain, called microglia, and I'm quite excited about them. And I hope at the end of my talk, you'll be excited about them too. So Sophie asked us to share with you a brief of our background and stuff, so I thought I'd start with my parents, who are the reason why I was born. So I'm Korean by ethnicity, so I was born in Indonesia. It's a long story, but I grew up in multiple cultures. I grew up in Korea, Brunei, and America as a teenager, and I went to college in the US. So my academic background and journey will be very different from what Selina and Dayne have shared with you. But I think the consensus of all our stories is that, as long as you have the curiosity and passion, you can become a scientist wherever you are in the world. So I grew up in different cultures and different, countries. But it was at college and when I became really curious about the brain. And it really started with me reading two books. One is a semi-autobiography called Tuesday with Morrie, and the other is by a really amazing neuroscientist called Oliver Sacks. He was a medical doctor. And I read this wonderful book that he wrote a while back. I'm sure this book is older than many of you here who are listening, and it's called The Man Who Mistook His Wife for a Hat. And these two books really inspired me and motivated me. And most of all, it made me really curious. And it made me curious about two questions. I became really intrigued by how does the brain work? And two, I became really heartbroken especially when I read Tuesdays with Morrie, who's describing his mentor who's dying of a very, very painful disease called Lou Gehrig's disease, or ALS. So I became really empowered to want to be part of this, right? How can we treat brain disease? And so I'm going to briefly describe my journey, and it's a long journey, but I want to emphasize to you that, what I'm studying today, I did not know when I was in college. When I was a student, when I was a college student, I did not know that I would be a brain scientist one day. So I want to make that point really clear. So when I was in college, I studied biochemistry. That is when I read these two books that really inspired me. So I just wanted to find out more. It was very vague. I just wanted to find out more about the brain, what neurobiology is, what neuroscience was. But that was when, through a lab that I joined as an undergraduate student back in University of Washington, Seattle, where I became really fascinated about these immune cells in the brain called microglia. So microglia act as the first responders of our brain. I'll tell you more about it later. Then I, stayed in the lab for about 3 years working as a technician, and that's where I began to become more and more interested in research. So for me, it took a while. I wasn't, Selina, she knew from secondary school that she wanted to go into science. For me, it took a while. I was like, no, I'm not really sure. And then I slowly grew to fall in love with neuroscience and neurobiological research. And it was during my PhD. So that was when I decided, okay, I think I want to study further. I think I really like this. So it was, after about 3 years working as a technician, I was like, I think I want to pursue PhD. So I went to study PhD in neuroscience at Harvard. And that was where I became really intrigued and by this really, really painful disease called Alzheimer's disease, which I remember reading in New York Times in 2010, a beautiful article that was describing this village where 2/3 of the population was basically dying of Alzheimer's disease, where you appear to be intact, but your mind is vanishing. And so after I finished my PhD studying Alzheimer's disease, then I did another training called a postdoctoral training, combining my interest in microglia, which I studied as an undergraduate student, as a technician, and Alzheimer's disease. And I was really interested in studying the role of these immune cells in dementia. And so with that, I started my lab in UK in 2018 in October. So I'm approaching 3 years coming to London. Describing very briefly, I know Nick did a wonderful description about Alzheimer's, and Selina also showed you some, images of the brain. So I just want to go a little bit more about the socioeconomic status of dementia, that it is not unique to First Nations, that dementia is actually a global problem. There's 45 million worldwide, and more than half of them are actually, half of the patients live in low and middle income countries. There are 10 million new cases diagnosed a year. That means there's one new case every 3 seconds. So it is a huge problem that we're dealing with. And I have to say, dementia impacts every family, because my dad, who, I showed multiple pictures of him, actually was diagnosed of Alzheimer's and passed away right as I was about to move to UK to start my lab. So this, research is, I've described to you my journey so far. It is both intellectual curiosity as well as a personal kind of motivation for me to study as well. So what are microglial cells? So microglial cells are these ones that you see in green. So as I already told you, they're the immune cells of the brain. And when you look at brains of human patients who had Alzheimer's disease, as well as animal models, including mouse models and rat models, you often see these A-beta plaques. And both Nick and Selina have shown you images of these plaques. So these plaques are made of this protein called A-beta, which Selina studies in a dish. So when you look at brains of patients and also animal models, you often see these plaques, but there are these cells that surround them. And the cells here are labeled in green, and they're microglial cells. And very interestingly, recent genetic studies, including those by John Hardy, who is a person that Selina mentioned in her talk before, him and other people have altogether have found that microglia may be the driving forces in modulating or mediating Alzheimer's disease pathogenesis. So one of the critical questions we're hoping to address in our Alzheimer's dementia field is to understand what roles do these immune cells play and how do they impair the brain, right? So what are microglial cells? So the next few slides, I'm going to give you some intro about what microglia are. As I've told you many times, they're the brain's immune cells. So here I'm showing you a beautiful image of a microglial cell here labeled in white, and they immediately go after things here labeled in red. These are neuronal cells that are dying, that they go target, and they pull it into their soma, what we call soma, so that they can degrade them. Right? Here's another beautiful image showing, and these are this video that I'm showing you right now is a video in intact mouse brains. The mouse is alive and it's under anesthesia. And so what they did was these mice are genetically engineered so that you can visualize the microglial cells here in white. And so what you can see in the very beginning is that you can see that microglial cells are very motile. And here, what the scientist is doing is to induce injury. So he's putting on a laser powers to induce injury. And what happens next is pretty amazing. Immediately, microglial cells, the process zoom in and they try to repair this injury. So this is what microglial cells do. This is a living example that shows you that microglia are rapidly responding to danger and injury. And they also phagocytose or eat dying cells and debris. In this experiment, what the researcher did is to shown here in red are neuronal cells. Here in green are microglial cells. So what the researcher did was to induce another type of laser injury, but here directly onto the neuron. And here I hope you can appreciate the microglial cell immediately take up this dying neuron up to its belly, similar to the image that I've shown you 2 slides ago. So this is what microglial cells do. They phagocytose or eat dying cells and debris. And now we know from many labs around the world that are studying microglia that microglia not only eat stuff, but they're actually doing multiple things, communicating with neurons who are, the major cells by which we retain our memory, collect our memory, and so forth. But what microglia do, even in the healthy brain, is that they sense changes of neuronal activity. When there's things to clear, I've shown you some examples where they can clear those debris. And they can also, what we call prune. They're like gardeners. So if you want to have a beautiful rose blooms at the end, a gardener needs to prune. So multiple labs have shown that microglia act as gardeners or sculptors of these neuronal synapses, which is something that Dayne is studying right now in his lab. And so in all of this, what microglia do is to listen. The microglia are always listening to changes in neuronal activity and neuronal function in the brain, and it employs different mechanisms to do these things. So how does this relate to Alzheimer's? So as I told you before, in Alzheimer's disease, genetic studies suggest that microglia are important, but what are they doing in the Alzheimer's brain? And what is happening in the Alzheimer's brain? Well, what we do know and have known for quite a while is that in Alzheimer's disease, neurons lose synapses. So synapses, as Dayne has beautifully explained to you, are how neurons communicate with each other. So synapses are these kind of blank space that you see where one neuron talks to the other neuron. And so in an Alzheimer's disease, in both patient brains and also in using mouse brains, here I'm showing you an example from mouse brains, where you can visualize synapses. Here are the synapses are shown in red and green. So red and green are marking both different compartments of the synapse. Green, shown here in green, is staining for a presynaptic marker, and shown here in red are dots that stain for postsynaptic markers. So when you look at a synapse, they're often colocalized to be yellow, as you can see here. So what I hope you can already appreciate from here is that when you use mouse models as well as in patient tissue of human brains, what you can see is that synapses are being lost. So this is a phenomenon that we knew that was going on for a while. So what we know from the other work from other labs is that what happens in these synapses, that probably is what induces these synapses to die, is that these A beta proteins, which later on then go on to build up these plaques that I've shown you before, they start accumulating in synapses. And so when I was a postdoc, what I helped identify is that these microglial cells, the immune cells of the brain, are shown to have engulfed this synaptic material. So this is an example that we did in using mice as experimental models. So when you challenge mice with A beta that are toxic, that accumulate in this Alzheimer's brain tissue, versus controls, microglia are shown to have engulfed a lot of this material. And so another thing that we found is that this microglial engulfment doesn't happen everywhere in the brain, but it actually happens in a very region-specific manner. So these two are areas of the brain called the hippocampus and the frontal cortex, two areas known to be highly involved in learning and memory. And what we've shown is that microglial protein called C1q is upregulated in these two areas, but not in control areas such as the striatum and cerebellum. And these increase of this microglial protein C1q is important because what we and other people, other labs before have shown is that this C1q, which is produced by microglial cells, actually work to tag. It's playing tag on synapses, telling microglia to come and engulf. So what we found is that microglia are engulfing synapses in Alzheimer's brain through this complement pathway. And very, very exciting for us is that when we blocked this pathway, this microglial pruning pathway, we could protect synapses and we could also protect memory in Alzheimer's mouse models. And one of the antibodies that we used to treat these Alzheimer's mouse models is actually in clinical trials right now to not yet for Alzheimer's patients. They're first of all trying to treat glaucoma, which is another neurodegenerative disease that is age-related as well. So one of the major questions that I'm asking my lab is then trying to understand whether we can identify these microglial cells that have gone quote unquote awry. Microglial cells are engulfing synapses when you're not supposed to. And this question is important because when you use general markers for microglia, such as shown here in green, this is a green what you see here in green is our microglia staining the hippocampus of a mouse. So you can see they're kind of everywhere. But what we know is that when microglia start engulfing stuff, for example, synapses in an Alzheimer's brain, that they upregulate certain markers, certain unique markers. But when you do these kind of challenge in the complement knockout mouse, which doesn't eat synapses, microglia do not turn on these markers. So now we have some idea of which markers are uniquely expressed when microglia start eating and start digesting the engulfed material. And so here are some other beautiful images that my lab is creating right now to see what other markers do microglia express when they're starting to engulf synapses. And here is showing another image where you can see microglia chock full of a marker for lysosomes, which are basically the gut of microglia. The activity of the lysosomes go up when they're digesting the tissue. So what is our long-term goal for this? So basically what we are proposing is that we to develop markers that are uniquely expressed by microglia as they're engulfing synapses. And the long-term goals would be to spot these microglial cells, to use them as biomarkers in living patients, and ultimately to target uniquely these microglial cells to save synapses from malfunctioning or from being eaten. And lastly, I wanted to say that these kind of microglial neuron interactions is important not only for Alzheimer's disease, but actually for overall brain health. And the reason is because now there are multiple publications from various labs around the world that suggest that if you manipulate microglial cells, or if microglial cells are activated, quote unquote, or non-activated, then it actually has detrimental consequences, leading to multiple disease pathogenesis. And this implication for microglia neuron interactions have been shown in mouse models or animal models of Alzheimer's disease, such as the one that I've shown you, FTD, which is another type of dementia, ALS, Parkinson's, glaucoma, Huntington's, as well as neurodevelopmental disorders. So the role of microglia and complement have been implicated in schizophrenia as well as autism, and another type of a detrimental neurodevelopmental disorder called spinal muscular atrophy. So this is what my lab studies, and hopefully what we study in our lab, and and many other labs are studying this neuroimmune interactions, will have an impact not only to study one disease, but multiple diseases across life spectrum. And I want to end with the people who are actually doing the work in my lab. And I also wanted to show you this, is that you don't have to do a PhD and then a postdoc and then have your own lab to do exciting science. You actually can be involved in doing research early on. So I didn't write the, now I also have 2 undergraduate students in the lab who are doing it. So actually there was also a high school student that came all the way from Singapore to learn about microglia in my lab. So if you're interested at any point in your career, I would say, contact a local, group leader or local PI who, if you're interested at all, and, you can start by shadowing in the lab to see what the lab is studying. And you can contact any lab that you'd be interested in to understand more about the science. So you can start this journey at any point of your career. And with that, I will be happy to take any questions with with Selina and Dayne.
Thank you so much, Soyon. So amazing to see just how active the microglia are, just watching them in the brain. That's really amazing. Before we go on to the Q&A session, we just have a few points of call, really. So the first is about the British Neuroscience Association prize draw, which you will know that at the beginning I said that there will be an opportunity to enter a prize draw for a year's membership to the British Neuroscience Association. And we're really lucky that we've been able to work with them. They have an amazing careers section on their website with a really comprehensive list of resources if you're interested in getting involved and learning more about dementia research, but also neuroscience in general. So we'll be linking that to you in an email coming up, hopefully tomorrow, should this recording be all good. And that will contain a list of resources and then also a link to a post-event questionnaire. It will take you 1 minute, and that will give you access to the British Neuroscience Association prize draw. So if you could please fill that out, it would really help us out, as we would like to repeat the event, hopefully in future. So, and then the second point of call is that this month is actually fittingly World Alzheimer's Month. So next Tuesday is World Alzheimer's Day. So if you follow the UK DRI on Twitter, and also check out the UK Dementia Research Institute website, there's loads of things going on, just raising awareness of dementia research, but also the patients that are involved in the research. And that World Alzheimer's Day is Tuesday the 21st. So keep an eye on all of the social networks for that, as well as the email that will be coming into your inbox. And thank you so much for attending the event for the second day. So on to the Q&A. So Selina, maybe we can start with you. So a really interesting question from Abdul Rahim, and apologies if I pronounce that incorrectly. With the neurons made in the lab, would it be possible for these to be used to repair atrophy in dementia patients in the future?
It's a great question. Yeah, thank you. It is really a good question. So I think one of the kind of advantages of the development of induced pluripotent stem cells, where we're making them from people's skin, is when we think about transplantation therapy, it removes one of the big issues, which is kind of rejection. Because in theory, and we get asked this often by patients, is, well, if you're making these great neurons in the lab, why can't you just put them back in and kind of repair the damage? So in a way, there's kind of potential there, because you could replace someone's neurons with their own neurons. However, the challenge in Alzheimer's disease is that the atrophy is throughout the brain. So particularly at the late stages of disease, as you will have seen from the images and probably from yesterday, the damage is really widespread. And so at the minute, the technology isn't advanced enough that we can replace those neurons and expect them to integrate into the circuitry in the way that they do kind of during development. However, here I'm speaking specifically about Alzheimer's disease. There are some trials that are happening in early stage Parkinson's disease to look at cell replacement, and it's slightly easier. I put that, I mean, of course it's not easy, but I put it in quotation marks, because in Parkinson's disease, there's a very kind of defined region of the brain that's affected in the early stages. So it's much easier to kind of conceptually think about replacing those cells rather than in Alzheimer's, where it would need to be replacement throughout the whole brain. But never say never. Let's see.
Thank you. And thanks for that question. So that kind of, I guess, segues nicely to Parkinson's. So we had a question from Phoenix, who asked, what causes the tremors in elderly patients as a result of Alzheimer's disease on a molecular or neurochemical level? And I guess that applies also to Parkinson's disease too. So maybe Dayne?
Sure, it's a really good question. It's one of the fundamental questions, and trying to assess what's going on in Parkinson's disease. It's in neurodegeneration, as I mentioned at the beginning, the brain is, even though we've done lots of research, it's still a bit of a mystery, but what we do know is that there's a lot of interaction and crosstalk. The reason that you get tremor in Parkinson's disease is because of the loss of that neurotransmitter, that chemical dopamine. But it's not just the loss of that particular neuron set and the loss of that particular dopamine chemical. The chemical itself has knock-on effects to other chemicals that are important to other sets of neurons. And so the loss of this particular chemical or neurotransmitter in dopamine actually causes a loss of control in the system. As I was mentioning to you before, everything in the body likes to remain homeostatic, wants to stay in that optimal zone. And so the whole system of the brain is very, whilst it is quite plastic and can move around, I mean, depending on stimuli, you can actually get them to come back to normal by the interplay of the different chemicals that are there. The loss of one really puts a strain on everything else and really causes the whole system to sort of collapse in on itself. So the tremor is caused by the loss of this neurotransmitter and ultimately the loss of those cells, but it's actually having a lot of knock-on effects, and those knock-on effects is actually what we're really trying to understand. The same is essentially true in Alzheimer's disease. You lose acetylcholine, and this is another neurotransmitter that's very important in balancing and controlling the system. Yeah, it's a really good question.
Really good question. Thank you for that one. Maybe one for Soyon. How did you block the C1q protein to prevent microglial phagocytosis of the synapses? Thank you.
Yeah, that's a very good question. So you can do it two ways. Well, in patients, obviously, one of the ways that we can do is to use antibodies that are specific against C1q. And so when we apply that, we can prevent microglial engulfment of synapses. So that is one way. If you are using animal models to study more about the mechanisms, then what you could do is to genetically engineer that microglial cells do not express C1q. So right now there's two ways. Now there are more other ways that that the field is developing. And for example, I think it's something that Nick Fox was discussing yesterday about genetically targeting. So what that means is to use very small molecules such as ASOs or siRNAs, that's what they're called, to really go into the cell and then specifically at part of the DNA that where C1q is transcribed, and to block that. So there's multiple ways.
You're on mute, Sophie.
Thank you. And maybe one we can for all panelists today. So one question at the top here: what do the Alzheimer's genes do, and how is the cell's phenotype changed, and how does that lead to a change that induces Alzheimer's? I think you've all worked with Alzheimer's risk genes, so how does that affect your work and how the cells are changed?
Well, I'm the one who's unmuted. Thanks, everyone. leaving me with this tough question. I think, it's a brilliant question, and it's one I get asked a lot, and it's really embarrassing because we actually don't have a good understanding of what the normal function of some of these genes are, particularly the ones that cause the rare familial forms of dementia that I specifically work on. So we have quite a good understanding of what happens when there are mutations and when things go wrong. And the consequence of those mutations is that it encourages the production of this A beta peptide, which then builds up in the cells. And we think, I'm giving a very kind of 10-second answer here just for time, but we basically think the accumulation of this protein is somehow toxic, whether that's because it's overproduced or because the clearance is disrupted. However, we also know that, cells don't waste time doing things that they don't have to because they need to be efficient. So there must be a physiological reason that this cleavage and this production of A beta can happen as well. And that's something that we don't have a very good understanding of. There are some hints that perhaps it's useful for synaptic function, but actually, in spite of decades of research, although we've got a pretty good understanding of things going wrong, I don't think we've got a very good understanding of all of what the normal kind of healthy function of these proteins and these genes are. Maybe I'm going to hear differently from Soyon and Dayne.
No, I think that's fantastic. I think, this is a very multi-million-dollar question right now that is really important to answer. So I think, it's an excellent question. So, what we're beginning to find out is, which I try to emphasize in my talk, is that recent genetic studies implicate more than half of them are actually on these immune cells, expressed by these immune cells. So that is, something I keep in my mind, every day as we're studying, is how do these risk factors, these risk genes, change the function of microglia. And I see here a question about, working with animal models. Is it ethical? Is it unethical? And stuff like that. And let me tell you, as someone who has been working with animal models for a long time, for about 20 years now, it is hard. And I don't do it in a lighthearted manner. I take into account in every animal, if the experiment is necessary or not. And I have to say, for someone who's studying, interaction between immune cells and nervous cells, those things are really hard to recapitulate in a dish. So it is actually necessary for us to study these kind of neuroimmune interactions in animals, and that is the only reason why we're doing it. It's not because we like to do it. We don't, but that is the only way that will help us to understand how does deleting one gene that it has been shown to increase the risk for dementia, how does that change microglia? How does that change microglial's communication with neurons? So, I'm addressing two questions with my answer right now.
It's really well done. I think just to chime in there is I completely agree with Soyon's assessment. It's not something that we do lightheartedly, and we are striving to consistently try to replace that. But it's got to be understood that, for instance, the stem cell technology, as Selina mentioned, is a relatively new technology, and we're only beginning to understand that and how to utilize it correctly. As we progress and as we try to get to stages where we can actually really understand it, it will really replace it, because it would provide a really good model for a human disease. But as yet, it's much better to have a much more complicated system that's more intact. And in doing so, and just to sort of segue back into the previous question, we don't, as I mentioned at the top of my talk, we don't understand absolutely everything. For instance, astrocytes that make up a lot of the brain is something that for a long time was just thought to just be there. It was just these cells that supported the neuron. But we're now understanding that they have their own particular roles, their own particular functions and features. And when we look at the genes that are actually associated with Parkinson's disease, ALS, etc., we find that sometimes they're expressed in these particular subsets of cells. Now we know that they are so important. We're now looking at those normal functions and how they're changed by the mutations in the genes. And so it adds an extra level of complexity, but an extra level of understanding as to how the brain works. So as we're driving towards understanding Parkinson's disease and Alzheimer's disease as a whole, it helps to have more and more of the baseline foundation understanding of how the brain actually operates and exactly what's going wrong. So it's one of those things that I'm dating myself, the $64 million question. And in that, we have to understand that question is beautiful. It's brilliant because this is exactly what we're asking every single day. And once you guys work it out, because I'm sure you're going to become the neuroscientists of tomorrow, then you will have the building blocks for a cure and a therapeutic.
Thank you, everyone. That was really, really great. Selina, do you want to add to the question about ethics, which I think was brought up and was a really important point to bring up as well?
I can. Maybe I just add a comment to the discussion about animal models as well, which, I fully echo. And I think, as Dayne said, the technology of using human stem cells is new. And one of the other questions in the chat was, will we ever be able to grow a full brain? And we're very far away from that. But I would say that the models are becoming more sophisticated. So we're now moving to 3-dimensional models. We're looking at ways to kind of incorporate the different cell types. It's by no means perfect, but we are trying to become kind of more and more sophisticated. With regards to ethics, actually, again, induced pluripotent stem cells have really been transformative, because before they were an option to us, the main source of stem cells was human embryonic stem cells. And so these are cells which are isolated from early stage embryos, typically ones that were generated for IVF, but were surplus to requirements. But in order to extract the embryonic stem cells, it requires destruction of the embryo. And so that faced, significant ethical challenges, particularly, dependent on religious beliefs, whether you felt that destruction of the embryo is an appropriate thing to do. And because we're generating induced pluripotent stem cells from the skin, the patients that we ask to donate are able to consent to that process. So we can explain to them, we will take your skin, this is exactly what we will do with it. And they are able to say, yep, we agree to participate in that research. So it really does help to remove a lot of the ethical issues. I'll keep it short in the interest of time, but I would say that, every model we use, we have a responsibility to use it appropriately. So nothing is perfect, but actually it does kind of alleviate us of some of the major barriers associated with embryonic stem cells.
Thank you. And then there's this one question that has come up a few times. What is the driving force that increases synaptic activity while feeding?
That's a very multifaceted question, which is a good question. There probably are no bad questions, but it is also a very good question. To relate back to my fantastic colleagues and their brilliant talks, there are multiple different things that can change how a synapse functions. It can come down to those small building blocks that Selina was talking about, in that there are certain proteins that move those packets of chemicals, neurotransmitters, from one place in the synapse to another. So it moves it from what is called the reserve pool. So I always think of it in the back seat to the readily releasable pool. So the driving seats are right towards the front to be ready to be released. And so changes in how much of that protein is present, or indeed how much of it is activated, can change the driving force. You can also have extrinsic factors. So you can have things like microglia that Dr. Hong was talking about that can actually prune synapses and modulate their activity. It can actually remove things from the surface of those synapses, as she was talking about, which can actually alter the activity there as well. But one major thing that actually changes the driving force, and I haven't touched on multiple other things, but one of the major driving forces is something as critical as calcium. Calcium is this signaling mechanism inside cells, and actually release of calcium within these cells actually causes changes in how much these synapses are actually going to fire. And I think that it's quite interesting to think of this, everyday substance that your mum and dad gets you to drink in cereal and with your breakfast, etc. But calcium in itself has all of these different functions inside the cell. It helps in fertilization of ova, it helps in bone synthesis, it helps in driving synaptic activity. But it seems to have, what's really interesting is that it seems to have these this way of being released almost globally within the cell, but also can be released just in the subcompartments, which we're only again just starting to understand how it's capable of doing that. And in understanding that, it really will help us to understand how the synapse really works. So there's a lot of different functions that kind of marry up. I haven't even covered half of them, but they are a lot of things as we're starting to understand the building blocks of synapse as to how it can actually alter the driving force. But genes do really help us to understand where to look in the first place. And the mutations in the genes helped us to narrow down exactly what to look at in terms of disease in Parkinson's disease, Alzheimer's disease, etc.
Okay, so thank you so much for your questions. I'm sorry we couldn't get to all of them today, but we really appreciate you taking the time this afternoon to join us for Making It Brain. As I mentioned, the British Neuroscience Association prize draw will be sent to you. All you have to do is fill out a quick 1-minute questionnaire about the event, and that should be sent to you tomorrow. And this event is being recorded, and you will also be able to watch Professor Nick Fox and Dr. Sarah Marcy's talks from yesterday as well too, on those recordings. So all that's left to say is thank you so much to our speakers for giving up their time today. And I think that was a really interesting discussion. We covered so many different parts of dementia research, even from the ethics to the more molecular mechanisms, and it's been such a pleasure to host this event as well. So thank you all for attending, and we hope to repeat the event next year. So all the best to you for attending, and for your studies coming up as well. I know it's been a really tricky year, but hopefully this has given you a really interesting insight into dementia research. And yeah, that's all to say. So we will end the event now, and thank you very much to our speakers, Selina, Soyon and Dayne. Bye everyone.
Thank you, bye.